Welcome to mark scheme and commentary for the task book for biochemistry and cell organisation, namely enzymes question 8. At least it looks easier than the last question. Glucose oxidase is an enzyme. Here we are, so that's our enzyme there. That's our enzyme. Shown below, the normal substrate is glucose. That's our substrate, our enzyme substrate. And this other thing, arabinose, never heard of that. That's an inhibitor. Okay, enzyme substrate inhibitor. Name the class of molecule. Okay, it's kind of like the family of molecules. So go back to your biochemistry, and is it a carbohydrate, fat, protein, that sort of thing? So it's actually a protein in it. So enzymes are proteins. So a globular protein, but it just wants the class, so that's fine. On the diagram, label the position of the enzyme's active site. Okay. Active site. So think about your diagrams and your jigsaws. So where can we fit our substrate in? So our shape here is similar to that. Okay, so that's going to be our. What does it want us to do? Label the active site. So I'm going to put there active site. Or I could even label it like that sort of thing. As long as you're clear it's there. Explain why you chose that position, right? So we need something better than just it fits or it's the same shape. We need to describe this using proper biology words. So the proper biology word to describe the fit there is complementary. So we can say the enzyme active site shape is complementary. I'm nearly going to spell that wrong. Complementary to the glucose substrate. Okay, and then we get 2 here, so we just need to explain that. So we're going to say the shape similar or complementary are glucose will fit or bind to the active site. We could even say here it will form that enzyme substrate complex. Enzyme substrate complex. So I think we're fine there. Go back to my red pen. Explain how arabinose inhibits the activity of glucose oxidase. So how does this inhibit the normal glucose going into the enzyme? Well, the inhibitor also has this shape, which is also complementary to our substrate. And so in the world of chemistry, these are all just in kinetic energy moving around and colliding with each other. And if a substrate collides with the active site, we're going to get our product. If an inhibitor collides with our active site, it's just going to occupy the active site for a while, then maybe move away. But whilst that's in the active site, our substrate cannot bind with the active site. So that's going to be our answer. And this is going to be the inhibitor competing with the substrate. So this is going to be our competitive inhibition. So the competitive inhibition, and we need to say 4 things about it. So I'm going to say competitive inhibition. Hopefully you can write neater than me, and then the examiner can actually read what you put. So the inhibitor enters the active site or occupies the active site. Why is that important? That's because it blocks the active site. And if the inhibitor is blocking the active site, then the substrate or glucose, so glucose cannot enter the active site. So we're going to get less enzyme substrate complexes formed, and so we get less product formed. So all those will get your mark, and you just need to hit 4 of those. And then a little bit of a stinker at the end. If a person drinks methylated spirits, not a good idea. Blindness can result because the body turns the meths into this horrible chemical here. It's poisonous. One form of treatment is for the patient to drink alcohol. That sounds alright. Which is a molecule of similar structure. There we are, that's the clue. It's an enzyme question, and they're mentioning similar structure. Suggest how this treatment will reduce the toxic effect of the meths. Well, it's ringing a bell, similar structure, that's going to... We've already talked about similar structures and inhibition, and it's just talking about the same thing. So, but what is our inhibitor here? Well, this is our substrate, and that's our toxic product. Substrate, toxic product, and if we add this to the mix, that's our inhibitor. So our ethanol is an inhibitor. What type of inhibitor it is? It's a competitive. So it's a competitive inhibitor because of that complementary shape and so on. And so we actually do need to answer the question here. So how will this reduce toxic effects? Well, that means that our substrate can't be turned into our toxic product as fast because we are inhibiting it. So our meths can't be converted to our toxic formaldehyde as fast. So we are reducing the effects of this toxic product. So our bodies can deal with the product much better because it's being produced more slowly. We can break it down. So we need to put that into words. The toxic, the meths can't be converted to our toxic, what was it, formaldehyde. So it can't be converted to toxic formaldehyde as quickly. So that's going to be safer. So less toxic effects. So pretty tricky that question and the previous question. Any problems, you need to go to sport. All the teachers will help you. Don't be sheepish about going to sport. Well done.